రైట్, అందరికీ నమస్కారం. ప్రభుత్వ మహిళా డిగ్రీ కళాశాల జగిత్యాల, అదేవిధంగా గవర్నమెంట్ ఆర్ట్స్ అండ్ సైన్స్ కాలేజ్ కరీంనగర్, ఆర్బీవీఆర్ఆర్ ఉమెన్స్ కాలేజ్ అటానమస్ హైదరాబాద్, వీటితో పాటుగా టీజీసీజీటీఏ తెలంగాణ, వీరందరూ కలిసి సంయుక్తంగా మనకి అందిస్తున్నటువంటి ఉచిత ఆన్లైన్ కోచింగ్ బాటనీ. My dear friends, utilize this wonderful opportunity. So our CPGET entrance examination, botany, is on 19th August. So my dear friends, don't waste your wonderful opportunity and also utilize each and every second fruitfully. మనం ఇప్పుడు భారత వజ్రోత్సవాలు జరుపుకుంటున్నాం, స్వాతంత్ర వజ్రోత్సవాలు. మరి ఒక పక్క మన భారత దేశానికి సంబంధించినటువంటి దేశభక్తిని చాటుతూనే అట్ ద సేమ్ టైం పోటీ ప్రపంచంలో కూడా మన సత్తా చూపించాల్సినటువంటి అవసరం ఉంది. ముఖ్యంగా సిపిగెట్ 2022 ప్రిపేర్ అయ్యేటువంటి విద్యార్థులందరికీ కూడా హృదయపూర్వకమైన అభినందనలు. ఎక్కడ కూడా జంకు గుంకు లేకుండా మీరు విన్నటువంటి క్లాస్, మీరు ప్రాక్టీస్ చేసినటువంటి ప్రాక్టీస్ టెస్టులు, అసైన్మెంట్ టెస్టులు, అదేవిధంగా మీకు 15, 16, 17 3 మ్యాక్ టెస్టులు కూడా తయారు చేయడం జరిగింది. ఈ మ్యాక్ టెస్టులు కూడా మీరు ప్రాక్టీస్ చేయండి. వీటికి సంబంధించినటువంటి ఆన్సర్ కీలను మీ పర్సనల్ వాట్సాప్కి పంపించడం జరుగుతుంది. ఎందుకంటే సమ్ ఆఫ్ ది స్టూడెంట్స్ దే ఆర్ నాట్ రెస్పాండెడ్. సో ఎవరైతే మీ సిపిగెట్ హాల్ టికెట్, సిపిగెట్కి సంబంధించినటువంటి హాల్ టికెట్ 22 హాల్ టికెట్ పంపిస్తారో వాళ్ళకి వాట్సాప్లో వాటికి సంబంధించినటువంటి కీస్ను కూడా మేము పంపించడం జరుగుతుంది. మై డియర్ ఫ్రెండ్స్, ఆపర్చునిటీ. ఓకే, సో ఐ విష్ యూ గుడ్ లక్. మేడం, షెల్ వి స్టార్ట్?
Yes, sir. I am ready to.
Okay, today's our eminent speaker and resource person, one of the eminent faculty, Rani Madam Garu, Assistant Professor, Department of Botany, GDC, SIDDIPET Autonomous. Madam has huge experience in our plant sciences, especially botany. So today's one of the important areas and plant embryology. In that embryology, polyembryony is one of the important topic. So my dear friends, listen carefully during the class. If you have any doubts, you ask our respected resource person. Rani Madam Garu, please welcome and share your screen and start the session.
Okay, sir. Thank you, sir. Thank you very much.
ఏమో సార్ బ్యానర్ అయితే ఉన్నాయి కానీ మరి అది
Okay, good evening, my dear student friends. Today we are going to discuss about the polyembryony. So in this topic, just introduction, classification, origin of polyembryony, causes of polyembryony, and importance of polyembryony. So in this generally, the polyembryony means, the introduction purpose, the occurrence of more than one embryo in the seed is known as polyembryony. Okay, so simple, the occurrence of more than one embryo in the seed, that is known as polyembryony. So this phenomenon was initially discovered in the orange. So orange by Leeuwenhoek, Antony van Leeuwenhoek in 1790. Okay, so the polyembryony was first introduced or first discovered by Antony van Leeuwenhoek in 1719. After that, Ernest and Snarf. So in 1918 and 1929, they have classified into two types, Nana. So in the two, true and false polyembryony. One is the true polyembryony and the second one is the false polyembryony. So it's depending on whether the embryos arise in the same embryo sac or in different embryo sacs in the ovule. So it's depending on almost whether the embryos they are arise in the within the same embryo sac or in different embryo sac in the ovule. So it is the true, depends on true polyembryony and false polyembryony. polyembryony. Generally, the polyembryony is quite common among the conifers. So in the gymnosperms, it belongs to gymnosperms. It is quite common in the gymnosperms. But many species of both the dicotyledons and monocotyledons, they will be exhibits this phenomenon. So this is poly means many or more than one embryo in the seed. So that is the polyembryony. Generally, so it is the polyembryony. It is in the following, just to true and false polyembryony. So it's almost nest and snarf. As I told you that classified the polyembryony into two types, true polyembryony and false polyembryony. So in the true polyembryony, it may be subdivided into two types. So in that cleavage polyembryony and adventitive polyembryony. So almost in the true polyembryony, several embryos, they will be appear within the one embryo sac. So in other way, it is of four types, Nana: cleavage polyembryony and adventitive polyembryony. So development of embryo from other cells of embryo sac, including the egg, or many embryo sac of and ovules also giving rise to embryos. In this four, just we have to discuss the main types, that is the cleavage polyembryony and adventitive polyembryony. So in this, almost in the true polyembryony, the polyembryony may be whether the embryos arise within the embryo sac, within an embryo sac, either by a cleavage of the egg or from the synergids. So we know that complete the structure of the ovule, almost 3 antipodal cells, 2 synergids, 1 egg cell and 2 polar nucleus. So in this, almost in the cleavage polyembryony, where the embryos arise within an embryo sac, either by a cleavage of the egg or from the synergids or antipodals or endosperm. So in this, almost the product embryos within or by projecting into a single embryo sac, that is termed that true polyembryony. So in simple way, the true polyembryony means the several embryos, they will be appear with the one embryo sac. True means original. So almost more than or many or several embryos appear with the one embryo sac only. So that is known as polyembryony. Okay. Almost you can observe here the diagrams in the embryogenic mass. So it is the growing the proembryos, that is the production of embryos within or by projecting into a single embryo sac, that is termed as a true polyembryony. So and the second one is the in this, it is two types, as I told you, that cleavage polyembryony and adventitive polyembryony. So in the cleavage polyembryony, where the embryos arise within an embryo sac, either by a cleavage of the egg or from the synergids or antipodals or endosperms. Just to in this diagram, so the first one is the normal embryo and the second one is the lateral division will takes place in that. So it will divided into new embryos. So that cleavage means one will be cleaved, it divided into 2 or 3. So the embryo, it is the suspensor, normal embryo sac will after the division, lateral division. So it is just gives rise to, one will gives rise to 3 new embryo sac. So that means where the embryos arise within an embryo sac, either by a cleavage of the egg cell or from the synergids, antipodals or endosperms. That is the cleavage polyembryony. And second one is the adventitive polyembryony. So in the adventitive polyembryony, where the embryos arise from almost from the tissues, they lying outside the embryo sac, where the embryos arise from the tissues. So it will be lying outside the embryo sac. Just means For example, the cells of the nucellus or the integuments are they will be, but generally they will come to lie within the embryo sac only. So that is the adventitive polyembryony. Almost, so in the adventitive, almost you know that the main example Rutaceae family. So in this, the citrus ovule in section showing the normal and nucellus adventitive embryos also. So this is the integument, nucellus. This one is the normal embryo. This is the nucellus embryo, and this is the endosperms embryo. So that means the embryos arise from the tissue, it lying outside the embryo sacs. So almost in this, the cleavage polyembryony, cleavage of the zygote, it is the simplest type of the polyembryony. Okay, so here the increase in the number of embryos is due to the cleavage of just to this one. The cleavage polyembryony is the simplest type of the polyembryony. So here is the increasing the number of the embryos is due to the cleavage of the zygote or embryo. So this type of polyembryony is quite common in the gymnosperms and some sporadic in the angiosperms also. So first the cleavage polyembryony, Jeffrey in 1895, Nana, he reported that the cleavage polyembryony in the Erythronium americanum. So the first plant, Erythronium americanum, first reported by Jeffrey in 1895. Okay, the cleavage polyembryony is first time reported by Jeffrey in 1895. In which plant? Erythronium americanum. Okay, here almost in this, here the zygote divides into form a small group of cells. So which continues to increase in volume, and outgrowths also they are given out as its lower end. That means which function later on as independent embryos. So the other example of some cleavage polyembryony are met within the nymphia and empetrum. So empetrum nigrum and nicotiana and some of the eulofia. These all are some examples of the cleavage polyembryony. Okay, so first time reported in Erythronium americanum, and some other examples like nymphia, advena, and empetrum nigrum, nicotiana rustica, and eulofia epidendria. So these all are the some other examples belongs to cleavage polyembryony. Just we'll discuss some nymphia advena. So there are twin embryos in nymphia advena. There are twin embryos which are considered to have originated by the splitting of a very young embryo. So a similar splitting of the embryos has also been reported in the empetrum nigrum also. In nicotiana rustica, so an ovule with 2 embryos has been reported, of which the smaller one arises as an outgrowth, so from the apex of the primary embryo. In the some members of the family Orchidaceae, so Orchidaceae also the cleavage polyembryony occurs frequently. Orchidaceae belongs to monocotyledonous, Nana. So in the monocotyledonous, almost the Orchidaceae family also we will observe the cleavage polyembryony. And some other there are also some eulofia epidermica. So the zygote divides irregularly to form a mass of cells, so of which the collagen cells gives rise to a multiple embryos. So in other variations, some other the filamentous proembryony becomes the branched, and each branch develops in an embryo. That is almost the cleavage polyembryony. Some origin of embryos from synergids or antipodal cells also. So the embryos may also be produced from other parts of the embryo sac, just like such as synergids and antipodal cells. In the most cases, the synergids become the egg-like. Synergids also, they can act just egg-like to form the embryos with or without fertilization. So in this, the twin embryos have been recorded in some almost peltrifilum and fragaria, so some nylas. So that means where the fertilization of the synergids by two male gametes that is found, but endosperm is not formed in this manner. Okay. So whereas in Crepis and Sagittaria, Poa, and some Aristolochia, these plants, the fertilization of the egg and one or two synergids is followed by the formation of the endosperm. In these plants, that means the production of embryos from antipodal cells also very rare. So the Shattuck in 1905, he reported such embryos in Ulmus americana. So that means the production of embryos from antipodal cells is absent, but to synergids and
excuse me, madam.
Other cells.
PPT is not movable, madam.
Oh, just wait, sir.
Only cleavage polyembryony is visible.
Okay. Just wait, sir. These are not in PPT, sir. Just I will give extra information regarding the cleavage polyembryony.
Please proceed. No problem.
Okay, sir. Okay. So that is the cleavage polyembryony, and the second one is the adventitive polyembryony. So in the adventitive polyembryony, in this, where the embryo sac arises from the tissue lying outside the embryo sac, as I told you, thus the cells of the nucellus are the integuments, but generally they come to lie within the embryo sac. So in the adventitive polyembryony also, it is origin of embryos from cells of the embryo sac other than the egg cell, or it is also formed by embryos arising from cells outside embryo sac. So please keen observe in these two ways: origin of embryos from cells of the embryo sac other than the egg, and in the second way, embryos arising from cells outside embryo sac. So in this way, almost the development of the embryos from the cells of the nucellus and integuments, they have been reported in the citrus, so Rutaceae family, in this Eugenia and Mangifera. These citrus, Eugenia and Mangifera, these three are the best examples for embryos arising from the cells outside embryo sac. Okay. In addition to these genera, some adventitive embryony also been reported in the Nigritella and Cyanella, Opentia, Ficus, Trillium. So these all are some other examples like belongs to the adventitive polyembryony. So almost the classification of polyembryony in this Ernest and Schnarf. So they classified the polyembryony into two types. In that, first is the true polyembryony. So in the true polyembryony, we'll discuss that the cleavage polyembryony and adventitive polyembryony, and false polyembryony. So second one is the false polyembryony. What is false polyembryony? Almost this type, it includes the cases in which two or more embryos are formed as a result of the development of the aposporic embryo sac. Just this type includes the cases in which two or more embryos are formed as result of the development of the aposporic embryo sac. So later, so in the false polyembryony, later on, some Gustafsson in 1946, he proposed that the term polyembryony should be restricted to those cases only in which two or more nucelli have the cases only, it will be applicable. So in this, each with its own embryo sac, they will be fused at an early stage. So Gustafsson, he also proposed that true polyembryony restricted to embryos. So it is derived from one or several embryo sacs found in the same nucellus. Almost, so the false polyembryony also, it is origin of embryos from other embryo sac in the ovule. So simply, false polyembryo means origin of embryos from other embryo sac in the ovule. So sometimes it is also occurs due to the presence of multiple embryo sacs within the ovule, or it may be arise from the derivatives of the same megaspore mother cell, or it may be arise from two or more megaspore mother cells, or it may be arise from nucellar cells, that means apospory. So last class we will discuss that the apomixis. So in the diplospory, apospory also there. So in these 3 or 4 cases, the false polyembryony is occur. Okay, what are they, Nana? The derivatives of the same megaspore mother cell. The polyembryony may arise from the derivatives of the same megaspore mother cell, or it may arise from 2 or more megaspore mother cells, or it may arise from nucellar cells. So in some juglans or citrus or pova and casuarina, the twin embryos. So almost citrus, casuarina, the twin embryos, they are arising different embryo sacs. Sometimes some nucellar cells also, cells present in the collagel region, they will be produced the additional embryo sacs. They are also found in the trifolium and atrophaxis. So these 3 or 4 cases, the polyembryony may occur. Okay. Similarly, the development of 2 or more embryo sacs in the nucellar tissue. So they has also been reported in the Fragaria grandifolia and some malus hyperhensis. So some almost the nucellar tissue in which way, Nana? The development of 2 or more embryo sacs in the nucellar tissue. So it has also been reported in Fragaria grandifolia, and second one malus hyperhensis. So in certain plants, the embryos are derived from different embryo sacs in the ovule. So the multiple embryos may also occur by the fusion of 2 or more ovules. So that means the fusion of the 2 or more ovules in the Rhizophora micronata and some Kinostoma phoetium. So in these 2 plants, almost the multiple embryos, they also occur by the fusion of 2 or more ovules. 2 or more ovules fuse with to form the false polyembryony. In this, which plants? Rhizophora micronata and Kinostoma phoetium. So in other several members of the family Loranthaceae, the well-developed ovules are not found. So, and instead of it, a group of sporogenous cells, they are also formed in the in this case. So this gives rise to many embryo sacs, of which only one mature and other disappear. Cells also will observe in this way. Okay. So, and in this, almost the origin of embryo from synergids are antipodal cells. The embryos may also be produced. So this is formation of embryo from antipodal cells. These 3 antipodal cells, it may be formed number of cells. So the embryos may also be produced from other parts of the embryo sac, such as synergids and antipodal cells. So in most cases, the synergids become the egg-like to form the embryos with or without fertilization. So this is the synergids embryo. This is the zygotic embryo, and other 2 embryos also will observe in this. So the synergids become egg-like to form the embryos with or without fertilization. The production of embryos from antipodal cells is very rare, as I told you that. So this is the origin of embryos. So that is endosperm cells in the tube. So in 1898 in Belanophora and Woodworth in Alnus and others have reported the embryos developed from the endosperm. So in endosperm also gives rise to a polyembryony. So embryo develops normally from the egg cell. And other case, the origin of embryos from cell outside embryo sac. So the embryos also developed from the cells of the nucellus and integuments. For example, citrus and mangifora. So whatever it may be, in almost in the polyembryony cases, most of the citrus means Rutaceae family members, they will show this type of the polyembryony. That is the best example. Origin of embryos from other embryo sac in the ovule. So sometimes the polyembryony occurs due to the presence of multiple embryo sac within the ovule. So they may be arise from, as I told you, that the derivatives of the same megaspore mother cell, or from 2 or more megaspore mother cell, or from nucellar cells. So what is the causes of the polyembryony? So in the polyembryony, significance or importance of the polyembryony, in this many theories have been reported to explain the occurrence of polyembryony by different workers at different times. So in this, some of the important theories are, as we'll discuss that. So in that, first is the necrohormone theory. So necrohormone theory, this theory has been proposed by Haberland. This theory is proposed by Haberland in 1921. So it indicates that the degenerating cells of the nucellus act as a source of the stimulus for the adjacent cells. So this theory, it indicates that the degenerating cells of the nucellus, it act as a source of the stimulus for the adjacent cells to divide and form the adventitive embryo. Okay, almost in this, it will indicate that some cells, any cell, so it degenerating cell of the nucellus, almost in the center part, some part is degenerating, and the degenerating tissue also act as a source of the some adjacent cells, so they will be divide and form adventitive embryo. Abburapu bahupindata laga manaki general almost the center part nucellus, it will be degenerating, and it is also act as just a source like the adjacent cells. It will be undergo the some divisions. So divisions takes place in that cells. They will be formed as a adventitive embryos. And in this, almost the occurrence of the two embryos in the necrohormone theory, the occurrence of two embryos in the Enothera. So Enothera has been regarded to be in support of this theory by Haberland. So it is proposed by Haberland for that purpose, the occurrence of the two embryos in which plant? Enothera ramarkayana. So Enothera has been regarded to be in support of this theory. This theory has also been supported by other workers also. And second theory, that is the hybridization theory. So according to this theory, the occurrence of multiple embryos is due to hybridization. Okay, so almost according to Kappet, K-A-P-P-E-R-T, Kappet in 1933, the polyembryony, that is a recessive hereditary character. So that is governed by a group of multiple genes. According to hybridization theory, so it is first opinion of that Kappet. So according to his, the polyembryony is a recessive hereditary characteristic. So it is governed by a group of multiple genes. In this, the recombination of genes, it will takes place during the process of hybridization and forming a single unit that gives rise to the multiple embryos. Okay, so almost according to Leroy, so he was Leroy was of opinion that polyembryony in mango was caused by one or more recessive genes. In the mango plant, Mangifera indica, so polyembryony in mango, it was caused by one or more recessive genes. So the mango has been originated in Eastern India, where only the monoembryonate cases are available in India. On the other hand, in the secondary centers of origin of Mangifera indica, So such as China, Philippines, Sudan, etc. So they will be exhibit polyembryonate cases. It is that the dominant genes are present in the species they found in Eastern India, primary center. So while the recessive genes are presenting in the form of secondary center. However, a few Indian varieties of mango, such as Bombay, Fazli, and Langra, they are also have that the polyembryonate cases. So the some monoembryonate condition in some species of citrus. So that is found in because of the synthesis and release of certain volatile and non-volatile embryogenic inhibitors. The ovules have that volatile and non-volatile embryogenic inhibitors. So that's why in citrus, the monoembryonic condition is occurring that. So which are not found in the ovules of the polyembryonate species. This is the hybridization theory. So what is the importance of polyembryony? So in the almost this phenomenon, it will place an important role in the plant breeding and horticulture. Okay, so significance and importance of polyembryony. The polyembryony phenomenon plays an important role in the plant breeding and horticulture. And the nucellar adventitive polyembryony is of great value in the horticulture. So that is also very important. Where the nucellar seedlings of citrus have been proved to be better clones of arched stalk than the cuttings. So some nucellar embryos are supposed to be free from diseases, and some propagation of the fruit trees such as citrus and mango, and other important like the application of adventitive embryos is also important for providing genetically uniform seedlings in the fruit trees. So it can be used for the development of homozygous diploid, some artificial production of these embryos from the eggs or synergids. So these all are some importance of the polyembryony. Okay. So on the other hand, the nucellar embryos, they will be supported to be free from diseases, and the nucellar seedlings also rejuvenate the vigor. So that is generally lost after continued cutting propagation. And some adventitive polyembryony is much useful in the propagation of the fruit trees such as citrus, mango, and the application of adventitive embryo is also important for providing genetically uniform seedlings in the fruit trees. So, and the haploids can be used for the development of homozygous diploids, so which are of much value in that way. So due to the practical value of haploids in plant breeding, so the methods have been recognized for the artificial production of these embryos from the eggs or synergids. So this is the importance and significance of the polyembryony. And so this is the today's topic. With this, I can conclude today's class. If any more doubts, please ask me. Hello, sir.
Yes, madam, really wonderful presentation. Excellent topic, madam. It is very useful. The all corners of top important points has
Thank you, sir.
Today's class. So our students are get benefited this class definitely without any bias. So thank you very much, madam, regarding of this class. You have given a wonderful lecture and also your valuable time for our students.
Thank you, sir.
Your efforts never ever forever use of regarding of our CPGET free online coaching, madam. Thank you very much.
Thank you, sir. Thank you.
Right.